Hello, am I audible? Hello, good evening to everyone. Hello, Mercy, how are you? Good evening. Okay, uh, yeah. Yesterday you were not there. Yesterday we have discussed one not too much things. In my first lecture, we have discussed basics of carbohydrate. Then we have done classification of carbohydrate in enantiomer, diastereomers, epimers. So up to this portion we have done in our first lectures, and then in my second lecture, I am good, Mercy. I am good. In my second lecture, I have talked only about anomeric carbon and anomers. If you have not have yesterday, then you can do one thing. You can watch it that video. Whenever you are having any kind of difficulty, you can ask me. I will repeat it. Fine. Now, we have stopped yesterday at this place. That we were talking about tautomeric structures. Tautomers are one kind of isomers only. It is also a stereoisomers, you can call it, where two or more than two different structures we are having for a compound, and that is on the basis of, means two structure differences is just only the hydrogen we are going to change their places. So if we will change the place of hydrogen from one carbon to the other carbon, so then we are going to call it tautomeric structures. So we have different types of tautomeric structures. Later on, probably if we will talk about nucleic acid. There also we are having some kind of tautomeric structures. In carbohydrate also we are going to see some kind of tautomeric structures. So different form of tautomeric structures we are having. Just for example, if you say it, we are not at all going to carbon skeleton change. So the main template, main carbon skeleton, main body of the compound is unchanged. Only the functional group will be changed, and that is on the basis of the change in the place of hydrogen. So if you will change the location of hydrogen, good evening Rajiv, how are you? Like you can see in this compound, this is ketonic group. So what we are going to do, this hydrogen we will change from here and we will put on this oxygen. So the location of hydrogen has been changed from here to hydrogen, and the double bond from hydrogen it will come here. So ultimately this here we will have a double bond. So if you see that these two structures, so this is previously was this is the ketonic structure, this is ketone. So this is called as keto form of a compound, and when we have changed hydrogen we have got this form, so alcohol and a double bond. So this is called as in, and this is called all. So this is overall called as in all structure. So we are having one kind of tautomeric form, that is called as keto enol tautomers. So whenever we are having a keto forms, if you are going to change hydrogen from one place, one carbon to the other carbon, and ultimately you will have this type of a structure, double bond and OH group, we will call it enol form. So it is called as keto enol tautomeric structures. Both can be interchangeable, means keto can give you enol form, enol can give you keto form, but the stability varies. So both the compounds will have different type of stability at a normal temperature, normal conditions. So if you will see a stability of these compounds, keto normally stable 99.9% cases you will have keto form exist. Enol forms exist very less, like that is 0.001% cases only we are having enol forms. What is the significance of this keto enol tautomeric structure in the case of carbohydrate is, when we will see carbohydrate, like this is my normal structure of glucose. I have written only 3 carbon, remaining 3 carbon is there, carbon number 4, 5, 6 I have not written, I am going to designate with the R. So in this case, this is ketonic type of group. We are having a double bond O and we are having OH. What we are going to do? We are going to change the place of hydrogen. So this hydrogen which you are going to see here, the place of this hydrogen will be changing to this oxygen, and this double bond will come here. So ultimately we will have this type of a structure. This O becomes OH and this H is not there, and we will have a double bond. So this is called a keto forms. This is keto form of compound, and this is in means double bond and OL all means alcohol. So this is enol form. Now, so glucose can be converted into a enol form that we are going to call enediol. Why we are going to call enediol, we are coming to later. So this is a kind of keto enol tautomeric form, tautomeric structure. So glucose can be converted to an enediol forms in alkaline condition. The condition should be alkaline. So this reaction takes place in alkaline environment. Now similarly, fructose can also be converted to enediol. Like this is keto structures. So from one of the hydrogen from here, this H will be coming at this O, and this double bond will go there. So ultimately you will have same structure. So either glucose or fructose or mannose, these three compounds normally going to convert it to enediol type of a structure. What is enediol? Till now we have seen only enol. We have seen enol form. En means double bond, all means alcohol form. What is enediol? Enediol means en means double bond and di means two, all means alcohol. Means both side of double bond we are having alcohol group. So this is called as enediol form. So glucose can be converted to enediol form in alkaline conditions. Fructose can also be converted to enediol forms. Mannose can also be converted to enediol forms in alkaline conditions. Now, if I want to convert glucose to fructose, or fructose to glucose, or mannose to glucose, or fructose to mannose, means any two carbohydrate, if you want to change it, if you want to convert it. So this conversion can be done via this enediol. Means first glucose will be converted to enediol forms, and then this enediol will be converted to fructose, or similarly fructose will be converted to enediol and enediol will be converted to glucose. So this is a tautomeric structure where in alkaline condition glucose, fructose, mannose can be interchanged from one compound to other compound, third compound to fourth compound. This reaction is a kind of transformation reaction where one group, one compound is transformed to other compound to the third compound. This reaction is named as Lobry-de-Bruyn-Vu-Enkestein transformations. If you able to remember it, it's perfectly fine. If you are not able to remember it, that is also perfectly fine. So this is a tautomeric structure. We have not done anything. What we have done it, we have just changes the place of hydrogen. In this case, I have changed this hydrogen from here to here, and this double bond will come from here to here. In this case, we have changed the location of this hydrogen from here to here and double bond from here to there. In this case, we have changed the location of this hydrogen from here to here and double bond from here to here. In all three cases, by these modifications, you will get this structure. Probably if you remember, whenever we are doing a Benedict test or failing test, that time our condition is alkaline. So in one case we are going to use sodium carbonate for alkaline condition. In another case we are using sodium hydroxide for alkaline condition. So both Benedict test and failing test occurs in alkaline condition, and there the actual reaction of carbohydrate or the any reducing sugar is through enediol formation only. So first their enediol formation will occur and then actual reactions happens. Is it clear? Is it clear, Merce? Rajiv, Ravina, is it clear? Now, fine. So next we are going to say the oxidation of glucose. We know in the glucose we have two kinds of structures, two kinds of functional group we are having. One functional group we are having, that is acid alcohol functional group, or we are having aldehyde group. These are the two functional group we are having in the glucose. So whenever we are going to do oxidations, both alcohol can be oxidized and aldehyde or ketonic. All the three compounds can be oxidized. The normal principle of organic chemistry is, if you are having alcohol, if you are going to do oxidation, so it will be converted to aldehydic structure. So alcohol after oxidations gives you aldehyde, and if you will oxidize aldehyde, you will get acid structure, COOH. So this is the term final place, means alcohol after oxidations gives you aldehyde, and aldehyde gives you acid. Similarly, if you are going to reverse way, acid after reductions gives you aldehyde, or aldehyde after reductions gives you alcohol. This is the normal principle of reaction. Now, in the glucose, what we have seen in our normal glucose is, just one second, let me rub it and then I will. In the normal glucose, we are having CHO, then we are having CH, like this one, and terminally we are having CH2OH. So if we are going to see it, we can have oxidation of alcohol also, we can have oxidation of aldehyde also. All the alcohols are not necessarily or preferably going for oxidation. Most preferred place which is going for oxidation is either this aldehyde group, which will go for oxidations, or this primary alcohol. So secondary alcohols means, if you are going further, secondary alcohol or tertiary alcohols normally not easily go for oxidations. Primary alcohol is better, easily they will go for oxidation. So here two places which is easily going to be oxidized, either the terminal aldehyde group, or this side, from other side it will be primary alcohol. Now if you are going to oxidize these two compounds, so we will get three different kind of products, three different kind of acids. Ultimate thing, either you are going to oxidize aldehyde structure, then also you will get acid. Final means ultimate oxidized product is acid. If you are going to oxidize aldehyde, then also final ultimate product will be acid only. Means in any case, either this will be oxidizing or this will be oxidizing. So ultimate product will be acid. Now, so we will have three different kind of compounds, three different kind of product we can get after the oxidations. So if only terminal CHO, the carbon number 1 is going for oxidation, means only CHO will oxidize and it will give you COOH, acid. So aldehyde after oxidation gives you acid. So if only first carbon is going for oxidation, we will get a acid. The name of that acid is called as gluconic acid. If I will tell, if only, I will repeat it, Tanuja. I have told in the carbohydrate structure, this is my carbohydrate structure. If you will see, it is having one terminal at first place, it is having aldehyde group, and the last place, carbon number 2, carbon number 3, carbon number 4, carbon number 5, carbon number 6, we are having alcohols. So either we are having secondary alcohol or we are having primary alcohol. So when we are going to see the oxidation of primary and secondary alcohol, preferably or preference or easiest way, primary alcohol is easy to oxidize compared to secondary and tertiary. So primary alcohol, that is the terminal 6th carbon alcohol, will easily it will go for oxidation. Now, so carbon number 2, 3, 4, 5, that is secondary alcohol, they are not going for oxidation. So mainly either carbon number 1 or carbon number 6 will go for oxidation. As an organic chemist, in the organic chemistry, if you are going to do oxidation of any alcohol, it will be converted to aldehyde, and aldehyde will be finally converted to acid. Means alcohol after repeated oxidations gives you acid. Similarly, alcohol or ketone after oxidation gives you acid. So ultimate result of oxidation of alcohol and aldehyde ketones is acid only. So if you are having ketone also, then acid, alcohol also, aldehyde also, then also acid only. So in all the three cases, either alcohol or aldehyde or ketone, we will get acid only. Now I have told you this carbon number 2 to carbon number 5 are normally not going for oxidation because they are secondary alcohol. So the carbon which will go for oxidation is aldehydic group or this primary alcohol group. So on the basis of which carbon you are going to oxidize, you will have three different kind of products. What are the products? If only terminal aldehydic carbon is going for oxidation, so if you will oxidize aldehyde, you will get acid. So this aldehyde after oxidation converted to acid. So this compound, the carbohydrate where only aldehydic group has been oxidized, we will get a product, we will get acid. We are going to call it D-gluconic acid. Why D-gluconic acid? Because this carbon is in the D form. So it is called as D-gluconic acid. If this aldehyde is not going for oxidation, only this primary alcohol will be oxidized, then we will have this COOH, this type of a structure, and this is called as D-glucuronic acid. So either D-gluconic acid or D-glucuronic acid when only one carbon will be oxidized. If aldehyde is oxidized, D-gluconic. If primary alcohol will be oxidized, D-glucuronic acid. And if both oxidizing at the same time, means this is also going to for oxidation and this is also going for oxidation in the same compound at the same time. So we will have here also we will have COOH and here also we will have COOH. So that structure we are going to call it glucosaccharic acid. So either you are having gluconic acid or glucuronic acid or glucosaccharic acids after the oxidation of glucose. So this is the glucose structure we are having. So either this will be oxidized or this will be oxidized. So we are going to get, if only this aldehyde will be oxidized, gluconic acid, and because this is D form, so it will be D-gluconic acid. If only this sixth carbon will be oxidized, we will get glucuronic acid because it is also D form, so it is also D-glucuronic acid. And if both this compound and this will be oxidized at the same moment of time, we will call it glucosaccharic acid. So this is the oxidation product of glucose. Similar things happen in case of fructose also. Is it clear? Is it clear, Mercy, Shivangi, Rajiv, Ravina? Anyone else? Fine. Any question? If you are having any question, you can ask. You can type there. If you are having any question, you can type there. Otherwise, we will go for reduction. Fine. So whatever carbohydrate we are having, either in the straight chain or in the ring form, the carbohydrate we are having different places. We are having alcohol group. So alcohol position we are having at second place also. Here also we are alcohol. This is anomeric carbon. This is second carbon. This is third carbon. This is fourth carbon. This is fifth carbon. This is sixth carbon. Now, whenever we are having this type of a structure, the carbohydrate is having a tendency, it will react. OH is very functional group, it will react with some of the compound. So we can have different types of derivatives. So like if OH, here previously we had OH, if hydroxyl group is replaced by a amino group, so OH has been replaced with a amino group, we are going to call this is amino derivative of glucose, so it is called as glucosamine. So amino derivative of glucose is called as glucosamine. Similarly, this is amino derivative. In amino we are having acetyl group, CH3CO group is called as acetyl group, so this is N-acetyl group. So if OH is replaced with N-acetyl group, so it is called as N-acetyl glucosamine. Why alpha? Because this OH is downside on the anomeric carbon. If OH will be downside, it will call as alpha. Why D? Because this is coming from D-glucose. So these are different derivatives. Glucose can form, fructose can form different type of derivatives by reacting. The OH group will react. In the polysaccharides we will see different derivatives which will give you different polysaccharides. Now come for second reaction. Second important properties, characteristic feature of the monosaccharides are their reduction. Now again you are saying that in the glucose we had OH group. So I had told you, if you are going to oxidize this one, you will get aldehyde, and if aldehyde will be oxidized, you will get COOH. Now this will give you this one by reduction, and this will give you by reduction. Now in the glucose, either you have aldehyde or you have alcohol. So possibility, probability, because we don't have alcohol group, so we cannot reduce acid group. Acid we don't have in the glucose or the fructose, so we cannot reduce that acid group. Either we can reduce this one, or alcohol cannot be further reduced. So only one option we are having for the reduction, either aldehyde group or the ketonic group. So wherever glucose or fructose have either aldehyde or fructose, only that can be reduced, that will go for reduction. So whenever we are going to reduce either aldehyde or ketone, the ultimate thing is we will get corresponding alcohol. So alcohol will be the product. Now, so that is shown here. This is glucose. So we have not written 5 other carbons, carbon number 2, 3, 4, 5, 6 we have not written. Only first carbon has been written there, and if you are going to oxidize this aldehyde group, the product will be alcohol. So this is going to give you corresponding alcohol. So here primary alcohol will be the product. So that is written there. When treated with reducing sugar, reducing agent, any reducing agent which will reduce your sugar, carbohydrate, the aldehyde or the keto group of monosaccharide is reduced to corresponding alcohol, which is indicated here. So if you will see the examples, like if we are having D-glucose, so the first carbon CHO will be converted into CH2OH. The CHO of glucose has been converted to CH2OH, and this structure remaining thing will be same, only terminal CHO we have changed it, and that we are going to call it sorbitol. Probably when you if you have might have studied diabetes mellitus, in the diabetes mellitus this reaction occurs by aldose reductase enzyme, and that is going to lead to one of the complications, sorbitol. Sorbitol is having very hygroscopic nature, so it absorbs moisture from the surrounding. In our eye especially, it will collect water from the surrounding because it is having hygroscopic nature, and when water will be there, cell will swell up. Like for example, this is my cell which has this type of reaction. So glucose has been converted to sorbitol, and this sorbitol is hygroscopic. So what it will do, it will absorb moisture from the surrounding tissue, surrounding extracellular matrix or surrounding tissue. So all these cells will have too much amount of water, and if cell will have very high amount of water, cell will swell, cell will be more turgid, osmotic pressure will be very high, and the normal physiology, normal function of cells will be affected. Cell will not do their normal behavior, normal functions will be not performed by that cells. So that is one of the drawback of this conversion in our body. It causes complication of diabetes mellitus. Similarly, if we are having galactose, galactose can be converted to dulcitol, the reduced product of galactose. In galactose also terminal aldehyde will be converted to CH2OH, and remaining at the carbon number 4 we are having different structure that we have seen already. If you are having mannose, it will give you mannitol. If you are having ribose, it will give you ribitol. In the case of fructose, whenever we are having fructose, fructose is going to give you two kinds of product. Either it will give you mannitol, it will give you sorbitol. Why this happens? Why fructose gives you two different products? Can anyone will tell me why fructose give me two different products? So this is glucose. Just think it, and I am coming there. This is glucose. In the glucose, if this CHO aldehyde group is going for reduction, so I have told you aldehyde after reductions gives you primary alcohol or alcohol. So this will be converted to alcohol. So you will have primary alcohol, and this is called as sorbitol or commonly glucitol. More commonly we are going to call them sorbitol. Similarly, in case of galactose also everything will be same. At the fourth carbon, OH will be this side. So every product will be same. If I will put OH this side, it will be called as galactitol. Now the question which I have asked you about fructose. In the case of fructose, why we are having two product? Why we are having mannitol, or why we are having sorbitol? Can anyone will tell me? Can anyone tell me why fructose give you two product, mannitol and sorbitol both? Mercy, Tanuja, Rajiv, Shivangi, Ravina, anyone, can you tell me why fructose give you two different products, mannitol and sorbitol both? No. मर्सी बताएंगे? तनुजा आप बताइए. नहीं, चलिए ठीक है, कोई बात नहीं. This is my fructose. If you will see this fructose is having ketonic group. So we are having one carbon this side, and then we are having ketonic group, and then we are having this thing. So we are going to convert this CO into alcohol. So this ketonic group can be converted to CHOH. So this side also you are having carbon, this side also you are having carbon. No. So depending on how you are going to write this carbon, so you will have two structure. This is carbon number 2. So if you are going to write OH on the left hand side, if OH you are going to write on the left hand side, this side, that will be mannitol, and if OH will be on the right hand side, it will be sorbitol, because this is ketonic group. So if you are going to oxidize ketone, you are going to get CHOH. This COH can be written in two structure, two forms. Either it will be written in the form of COH and H, or it will be written in the form of CHOH. If you are going to write this type of a structure, right hand side OH, it is called as sorbitol, and if you are going to write OH on the left hand side, it will call mannitol. So probability is there, both type of compound will be forming. So fructose or any ketone can give you two types of alcohol, because ketonic group is present in the between of the chain. So whenever it will be going to give you product, I will tell you what I have told Ravina. In this case we are having ketonic group. So I have told कि whenever you are having aldehyde or ketone, if you are going to oxidize, reduce it, so reduction of aldehyde and ketone give you corresponding alcohol. So this carbon which is you are having this type of a structure, and this is the ketonic group. So this ketonic group is going to be converted into CHOH. Now, how you are going to write this CHOH? It can be written in two forms, either COH and then H, means OH I have written in the right hand side, or second form I can write OH on the left hand side. So this CHOH can be written in this form also, this structure also, means whenever you have converted, you have reduced your fructose, you have got CHOH, and this CHOH either in this structure or in this structure, both are possible. So whenever you are going to have this structure, right handed side OH, that we are going to call sorbitol. So if this C double bond O is going to oxidize and give you this type of product, right handed side OH, it will give you sorbitol. And if oxidation of this one give you another product where OH will be left hand side, it is called as mannitol. Means fructose of this carbon, just look at only this carbon, if this carbon after reduction gives you this type of a structure, it will be sorbitol. And if this carbon gives you this type of a structure, this is called as mannitol. So both possibilities are there, both type of compound can be formed. So that's why we are going to get two kind of alcohols when fructose will be reduced. Whereas in case of glucose, because it was primary alcohol, so possibility is, because primary alcohol is symmetric carbon, so only one type of a structure is possible. You don't have either D form or L form or right side or left side, everything will be same only. But in the case of this one, this will be asymmetric carbon. Both will form, percentage will be different, its sharing will be different. Means this will have some sharing or this will have some percentage, but both will form. This is depending on which, like we have seen alpha and beta form of anomeric structures, both are going to present only their contribution, their percentage sharing will be different, but both will be there. Clear? Clear, Mercy? Because the reduction of this compound gives you asymmetric carbon, so that's why you have two types of structure possibility, either D form or L form, irrespective to that carbon, and that's why we have two different compounds. Both will be going to form, only their contribution, their percentage will be different. Next property of primary means monosaccharides are called dehydration. Because whenever any compound will have more than one alcohol group, any compound which is having more than one alcohol group, OH group, it can go for dehydration reactions. So the carbohydrates, glucose also having multiple OH group, means it is polyhydroxy compound. So whenever we are going to have a polyhydroxy carbohydrate, and if you go take it for dehydration, you are going to dehydrate it, remove water molecules, so you will have a dehydrated product. The glucose is having almost 5 different OH group and one aldehyde group. So the maximum dehydration, what you can do it, you can remove 3 water molecules from the monosaccharides, hexose carbon. So for every 2 carbon, 1 water will be going out. How to do dehydrate it? In our compound, we are going to use mineral acids for dehydration. So we are having either HCl or HNO3, nitric acid, or H2SO4. These are called as mineral acids. These inorganic acids are very strong dehydrating agent. So whenever we will put these acids along with carbohydrate, it will go for dehydration. It will take my carbohydrate for dehydration. So water will be removed. Like this is my glucose. If I will add glucose, or glucose is going to be reacting with sulfuric acid or hydrochloric acid or nitric acid, means any strong acid, any dehydrating agent. So 3 molecule of water is going out, and ultimately it is going to give you a ring structure will form. That ring structure will be roughly like this one, and it will have 2 different compounds at different places. That structure I am going to show it to you. Means the carbohydrate monosaccharides are going for dehydration, and extreme dehydration gives you 3 molecule of water. When you are going to remove the product, we are going to call that is furfural derivative. What is furfural? So either you are having 5 carbon sugar or the 6 carbon sugar, both going to give you furfural derivatives. 5 carbon sugar also will leads to furfural, 6 carbon sugar also leads to furfural. Only difference is, in the case of 6 carbon sugar, if you are having hexose, you are going for dehydration, any hexose. That time this terminal aldehyde is not if you are having this hexose, you are going for dehydrations, that there will be a ring structure will form. So there this both the terminal carbon, either first aldehyde group and the 6th primary alcohol, will be not participating in the ring formation, and this type of structure is called as hydroxy methyl, and this is furfural. So this is 5 hydroxy methyl furfural. Why? This is 5th carbon. So at the 5th place we are having hydroxy methyl furfural. So this is furfural compound, furfural derivative, and at the 5th place we are having hydroxy methyl group. In the case of hexose, when we are having pentoese, that time we don't have any hydroxy methyl, we are just getting furfural. So just remember it, whenever we are having monosaccharides or the polyhydroxy carbohydrate reacts with any dehydrating agent, it gives me furfural derivatives, furfural compound. If we are having pentoese sugar, it will give me furfural. If we are having hexose sugar, it will give me a hydroxy methyl derivative of furfural, because at the 5th place this hydroxy methyl will be there. So if this is your pentoese, I have told you if it will react, it will give you a furfural compound. So any compound which is having this type of ring, 5-member ring with 2 double bond and 1 aldehyde here, this is called as furfural. So both pentoese and hexose gives you this derivative. Only difference is, in case of pentoese we don't have any substituent at 5th carbon. In the case of hexose we are having 5 hydroxy methyl derivative. So this is my dehydrated product. So after dehydrated product of pentoese is furfural compound, dehydrated product of hexose is hydroxy methyl furfural. Now this dehydration reactions we are going to use for a diagnostic test, for a detection test, for a qualitative test of carbohydrate. Whenever we want to detect carbohydrate in any test sample, we are going to do a Mollis test. What is Mollis test? In the case of Mollis test, we use just a carbo, that is a alpha naphthol. If we are having, if we don't have alpha naphthol, we can go for other compounds also, like thymol or other compounds, but preferably we are going for alpha naphthol. We are going to use alpha naphthol. So what we will have, in the case of Mollis test, first your carbohydrate will be dehydrated. If we are having pentoese sugar or hexose sugar or any other sugar, it will be dehydrated product. The dehydrated product will be either furfural or furfural derivative. This furfural or furfural derivative reacts with alpha naphthol, which we are having in the Mollis reagent. Naphthol in the Mollis reagent. So this furfural or furfural derivative combines with alpha naphthol and gives you a product, colored product. that is of purple color. So whenever we are performing a Mollis test, at the junction we can see a purple color ring form occurs. Ring is there. This purple color ring formation is because of a complex compound formed between alpha naphthol and furfural. If furfural or hydroxymethyl furfural will form, then along with naphthol it will give you complex and you will have a purple ring. If you don't have furfural, then no complex will form, so no ring is there. So you can detect, like in all the cases, which type of, whatever type of carbohydrate you will have. If you are having glucose, fructose or any monosaccharide, directly it will react. If you are having polysaccharides like a starch, so first those strong acid, whatever you have used, H2SO4, HNO3, HCl, those strong acid digest your polysaccharides into monosaccharides, and then monosaccharide will give you reactions. The reaction will be given by monosaccharides only. First polysaccharide will be converted to monosaccharide and then that will give you reactions. Similarly, in case of disaccharide also, the disaccharide will be first degraded into monosaccharides and then ultimately you will have a reactions. So this is a detective reactions, identification reactions which we do for carbohydrates, any form of carbohydrate, either monosaccharides, disaccharides, polysaccharides, free carbohydrate, combined carbohydrate, glycolipids, glycoproteins, any form of carbohydrates will give you positive Mollis test. So this is a positive identification test for any carbohydrate in any condition at any level. So this is sometimes, because we are using very strong acids. So when you are using very strong acid, we are using concentrated acid. So it will not take very much time, because strong acid is almost acid only. So it will break that bond. It is few seconds structure, few second time. But when we are going to compare with monosaccharides and polysaccharides, obviously polysaccharide will take slightly millisecond, microsecond more time. Compared to monosaccharide, polysaccharide will take more time, but that is not, we are not going to detect it. We are just going to use this test for the detection of the presence of carbohydrate is there or not. So this is all about carbohydrate. I have missed one of the properties which we will talk later on when we will have a separate properties we will talk, that is called as Osage test. So almost all the monosaccharides and disaccharides normally go for Osage test. Osage is nothing when your carbohydrate is going to react with a phenylhydrazine. Phenylhydrazine is a compound. Whenever your carbohydrate reacts with phenylhydrazine, it gives you a crystal type of a structure. So different compounds, different carbohydrates will give you different type of Osage test. So that we will talk later on in a separate test. Osage test we will have, and then we will talk about. So now we are going to see disaccharides. If you are having any questions in the monosaccharide, you can ask me. You can type there. Any questions on monosaccharides, if anyone is having, you can ask. No question? So disaccharides. Disaccharide is another form of carbohydrate where di means two and saccharide means units. So two units will join together by, and joining will be by a specific bonds. That bonds we are going to call it glycosidic bonds. We are going to see glycosidic bonds. So that is called disaccharides. Disaccharides are having some alternate names also. It is called as double sugar also. It is called as biose. Bi means two, os means sugar. So disaccharides have different names, but preferably we are using disaccharides only. Whenever two monosaccharides units, two simple sugar or simplest unit of carbohydrate joined together by a condensation reactions. What is condensation reactions? Condensation reaction is the combination of two units and you are going to remove water molecules. Polymerization you can call it, condensation you can call it, all both are same, only difference is removal of water or non removal of water. So it is a condensation reactions where water will removed, we will see later on. Similar to monosaccharides, disaccharides are soluble in water, they are sweet in taste. So monosaccharides and disaccharides both are called as sugar because of water solubility and their sweet taste. Both monosaccharides and disaccharides are called as known as sugar. The three common example we are using, there are a number of disaccharides we are having, but three common example what we use or what most of the people uses are sucrose, lactose and maltose. Sucrose is our common table sugar, whatever commonly we are using it. Lactose is a milk sugar and maltose is malt sugar. We will see all the three how it is going to form. So the difference in the disaccharides are due to their orientation, due to their arrangements. Whenever we eat, so my food contains polysaccharide also, disaccharide also. So I have told you we are having two monosaccharides, like this is monosaccharide A, monosaccharide B. These two monosaccharides will joined with a specific bond. So these two monosaccharide is going to break. If I will remove this bond, I will degrade this bond. So ultimately I will get two monosaccharides will be separated out. So A will be separated out, B will be separated out if you will degrade this bond. So I have told you we are having three common example of disaccharides like sucrose. So the enzyme which digests sucrose is called as sucrase, which digests the bond between the two monosaccharide of sucrose. We will say lactase. This is enzyme which digests lactose and gives you two monosaccharides. Similarly, maltase is the enzyme which digests the bond between monosaccharide of maltose and gives you two monosaccharides. So how this glycosidic bond forms? So two monosaccharides or more monosaccharides, monosaccharide to monosaccharide connection, the joining between two monosaccharides is done by glycosidic bond. In this glycosidic bond, how it forms? It is going to form, remember one thing, to form this glycosidic bond, one of the anomeric carbon is compulsory, means two thing is going to join. This is one compound, this is one another compound. So both are going to join together by a glycosidic bond. Most important thing is one of the carbons should be anomeric. Without any anomeric carbon involvement, no glycosidic bond will form. So minimum one anomeric carbon we need to form this glycosidic bond. So how it forms? It is going to form between the anomeric hydroxyl. So we are having, like this is the anomeric carbon, if you will see here. So this is hydroxyl group on the anomeric carbon, reacts with another hydroxyl group of another sugar. So means both will be hydroxyl group. So both places we are having hydroxyl group, OH group. This is from one sugar, this is from another sugar. Both are going to join together by the removal of water. So H, O and H will be removed. So ultimately you will have a bond with O will be left. If I will remove this OH and this H, so this is H2O. So H2O has been removed and only oxygen will be left. So that is the glycosidic bond. So ultimately you will have a glycosidic bond formation will be there. That we are going to see next slide. So first we will see how glycosidic bond forms and then we will see how to do its nomenclature. Now in this case, if you will say this is one of the monosaccharides, cyclic form of one of the monosaccharides, where this is the anomeric carbon and in this is the anomeric carbon. So both are anomeric carbon only. Now what I have told you, minimum one of the anomeric carbon we require. So here either we require minimum this one or this one. Minimum one is required. Maximum you can have both will be anomeric carbon, that is possible, but bond will be looking like ester only. Bonds will looks like ester only, but we are not calling ester bond, we are going to call glycosidic bond. So what happens in this case is, yes Mercy, bonds will looks like ester only. Okay, so now you can see here, in this case, this is hydroxyl group of one of the monosaccharide, which is going to join with hydroxyl group of another monosaccharide. So two monosaccharide is going to join together between by forming a bond. So both places hydroxyl group is going to participate, hydroxyl group is going to involve in the bond formation. In this case, minimum one anomeric carbon is required. If both will be anomeric carbon, I don't have any issue, but minimum one anomeric carbon I want. So this two is going to react by the removal of water. So water will remove this OH and this H will go, and ultimately one oxygen will be left out here. So this is a connection between these two monosaccharides by forming a glycosidic bond where oxygen will be in the center. This is called as O glycosidic bond. We can have N glycosidic bond also, but in the carbohydrate we are having O glycosidic bond. So we are not using O, we are just simple using glycosidic bonds. Now how to do nomenclature? Whenever we are doing nomenclature, I have told you minimum one anomeric carbon is required. So first monosaccharides, the first monosaccharide which we are going to use for the bond formation, that we are going to write anomeric carbon. So if I will use only one anomeric carbon of this sugar, so this will be my first carbohydrate. So I will write this carbohydrate here and this carbohydrate there. Means if you are using both anomeric carbon, then I don't have any issue. You can write any carbohydrate anywhere. But if you are going to use only one anomeric carbon, then I will write the sugar whose anomeric carbon I am going to use will be at the first place. Now this carbohydrate is alpha type or beta type. Can you tell me? This is anomeric carbon, and whenever OH will be below the plane, what we are going to call them? Alpha or beta. We have talked about in the anomeric structure anomers. So when OH will be below the plane, it is alpha type or beta type. Who will tell me? Anyone? If we are going to use OH, if here OH is below the plane and this is also anomeric carbon, so these two are alpha type or beta type. No, Anjali. Whenever OH will be below the plane, we are going to call them alpha. This is also alpha type and this is also alpha type only. So both are alpha type only. Don't guess. So how to write the nomenclature? How to do its nomenclature? So first we are going to write the type of carbohydrate. It is alpha type or beta type. So think I am going to use this anomeric carbon. So this is alpha type. So I will write alpha. Then I will write the carbon number. So this is carbon number 1 because this is anomeric carbon, that is carbon number 1. Remember always in case of glucose, carbon number 1 is anomeric. So this is 1. And then this alpha 1 is going to form a bond with second number of carbon. So it is alpha 1 2, and then because this is also anomeric carbon, so you can write its type. This is also alpha type only, so it is called as alpha 1 to alpha glycosidic bond. So this glycosidic bond is alpha 1 to alpha glycosidic bond. So first you have to write the type of anomeric carbon, then write the number of carbon atom, which carbon you are going to use, and then write with whom it is going to form a bond, and then write their type if it is anomeric. If it is not anomeric carbon, then no need to write this alpha or beta. Clear? So this is a glycosidic bond, alpha glycosidic bond, alpha 1 to glycosidic bond. Always remember, in case of glucose, the C1 is anomeric, and in case of fructose, C2 is anomeric. So remember that one, because it will help you many places. Now this is also, and this is anomeric carbon, and this is anomeric carbon, this two. Both the anomeric carbon I have used here for the bond formation. So I have used both anomeric carbon for bond formation. Clear? Now in this disaccharide, this is my disaccharide, where one monosaccharide joined with another monosaccharide by combining their two anomeric carbon. So both anomeric carbon I have used of this monosaccharide also and this monosaccharide also. So in this disaccharide we don't have any anomeric carbon. No anomeric carbon is free. In this disaccharide we don't have any anomeric carbon free. So whenever you don't have any free anomeric carbon in any disaccharides, we are going to call them non-reducing sugar. If any carbohydrate, if you don't have any anomeric carbon left out, if you have used both the anomeric carbon for the bond formation, we are going to call them non-reducing sugar. Fine. If in the same carbohydrate only, if the bond formation will not occur with by second carbon, bond will form with any other carbon. This fourth carbon is going to form a bond. So there will be a bond formation between this OH and this OH. So then it will be alpha 1 4 glycosidic bond, and in that case this anomeric carbon will be freely available, because bond formation happened between first and 4. Then this anomeric carbon will be freely available. So that time, if this minimum one anomeric carbon is freely available in the compound, then we are going to call them reducing sugar. So normal definition is, in any carbohydrate, if free aldehyde or free ketonic group is there, and remember the free aldehyde and ketonic group, aldehyde and ketone only is the anomeric carbon. This is ketonic group, this is aldehydic group. So if whenever you are having a disaccharide, look at anomeric carbon is free or not. If anomeric carbon is freely available, minimum 1, it will be reducing. If no anomeric carbon is free available, it is non-reducing sugar. Clear? I think it is clear for you. So we are going to have classification on the basis of those free reducing sugar, means reducing sugar or non-reducing sugar. It is, so we are having reducing disaccharide and non-reducing disaccharide, depending on the any anomeric carbon is freely available or freely not available. Like these are different examples of disaccharides. Like we have sucrose, which is also called as table sugar, cane sugar, beet sugar or saccharose. There sucrose is the combination product of glucose plus fructose. from glucose we are going to use first carbon, and from fructose we are going to use second carbon. Remember, I have told you, in glucose C1 is anomeric, in fructose C2 is anomeric. So if you are using first carbon of glucose and second carbon of fructose, means you have used both anomeric carbon. You have used this anomeric carbon and this anomeric carbon. C1 is anomeric carbon of glucose, C2 anomeric carbon of fructose. So if you have used both anomeric carbon, means no anomeric carbon has been left there. So when you don't have any anomeric carbon left, it is called as non-reducing sugar. So sucrose is non-reducing sugar. Why? Because no freely available any anomeric carbon is there in the disaccharide. Similarly, if you go for lactose or the bond formation between galactose and fructose. Galactose is similar to glucose only. So here also, in case of galactose also, C1 is anomeric. So we have used anomeric carbon of galactose, but from the fructose we are going to use fourth carbon, not the carbon number 2. So we are going to use C1 and C4, but this is not the anomeric carbon. Means after the bond formation in the lactulose, one of the anomeric carbon of fructose is freely available. Bond formation occurs between anomeric carbon of galactose and non-anomeric carbon of fructose. So anomeric carbon of fructose is free. It means this compound is, lactulose is my reducing sugar. Similarly, if you go for lactose, here also glucose and galactose. So in glucose also C1 is anomeric, galactose also C1 is anomeric, but we are not using both the places C1. So in case of glucose we are using C4. So means C1 anomeric carbon is freely available of the glucose. So one of the anomeric carbon is free. Means this is also reducing. Maltose, there also 1 and 4 we are going to use. So anomeric is free, it is reducing. Trihalose, we are going to use carbon number 1 of this glucose and carbon number 1 of this glucose. Means both anomeric carbon we are going to use. Means it is reducing or non-reducing. Trihalose, because we are using both the anomeric carbon C1 and C1. You can see here alpha 1, alpha 1. So when you have used both anomeric carbon for bond formations, will it be reducing or non-reducing? It will be reducing or non-reducing. Trihalose. Trihalose is non-reducing. Why? Because we have already used both the anomeric carbon. So no anomeric carbon is freely available in the disaccharide. We will see a structure also. Similarly, in case of cellobiose, in cellobiose we are going to use carbon number 1 and carbon number 4. So means glucose and glucose, only one anomeric carbon we have used. One is free already. So it is reducing. Ketobiose, we are going to use carbon number 1 and 4, means one anomeric carbon is freely available. So it is again reducing. So just look at the anomeric carbon is free or it is it is freely available or it is involved in the bond formation. If it is free, it will be reducing. If it is involved in the bond formations, then it is non-reducing sugar. So we are having some of the disaccharides, like we can see here. So we will go for first maltose, we will see, and then you can identify easily. So maltose is known as malt sugar or maltobiose. It is reducing sugar. Why it is reducing sugar? We will see. If you will see, this is the alpha D glucose and this is alpha D glucose. Maltose is going to form a bond between alpha 1,6 glycosidic bond. Means this is carbon number 1 and this is 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. In case of maltose, the bond is going to form between carbon number 1 and carbon number 6. So this is my carbon number 1 and this is carbon number 6. So there will be a bond between, in case of maltose we are having alpha 1 4 glycosidic bond. In case of isomaltose we have alpha 1 6 linkage, that we will see later on. Maltose we are having alpha 1 4 glycosidic bond. So this is first carbon which is of alpha type and this is second glucose, so bond should be alpha 1 4. So this is the fourth carbon. So there will be a bond formation between these two carbon by removal of water. So water will go and there will be a bond formation occurs by that is glycosidic bonds. Now you can say in this case this is the anomeric carbon, this is anomeric carbon and this is the anomeric carbon, but the bond is going to form between this 1 and 4. So one of the anomeric carbon is still free. So when any anomeric carbon is still free in the whole disaccharides, it means this carbohydrate, this disaccharide is reducing sugar. So maltose is a reducing sugar, which you can see here. The bond is formed between carbon number 1 and carbon number 4. One anomeric carbon is still free. This is anomeric carbon and it is free. So this maltose is reducing sugar and the bond is formed between alpha 1 4 glycosidic bond. Similarly, in case of cellobiose, if you will see it, this is carbon number 1 and carbon number 4 is going to form a bond. This is alpha type, this is OH will be here because bond you can see H is here, so OH will be here. In this case OH will be on the top. So this is beta type of sugar. This is also beta type of sugar. Why it is beta? Because OH is on the top. So when OH will be on the top, it is called as beta. Here also beta, here also beta. Now this bond is, this is carbon number 1 is going to form a bond with carbon number 4. So it is beta 1 4 glycosidic bond. Why beta 1 4? Because this carbon, anomeric carbon, is beta type. So we will write the type of sugar, then carbon number, then with whom it is going to form a bond, and if it is anomeric carbon then I will write alpha or beta. If it is non-anomeric then no need to write it. So I have not written here. So it is beta 1 4 glycosidic bond. In this case also this anomeric carbon is still free, means this is also a reducing sugar. So cellobiose is also a reducing sugar. So maltose and cellobiose both are going to give you glucose plus glucose, but in case of maltose it was alpha D glucose plus alpha D glucose. In case of cellobiose it is beta D glucose and beta D glucose. The bond will be alpha 1 4 in case of maltose and beta D glucose in case of cellobiose. In both the cases one of the anomeric carbon is free. So both are reducing sugar. Clear? Is it clear? Tanuja, Ravina, Anjali, Shivangi, Mercy, Umar, Rajiv, is it clear? Any confusion, anything, any question you are having? Mercy, any question you have? Okay, now we are going to say sucrose. In case of sucrose, sucrose is commonly we are going to call a table sugar, and this is most commonly we are going to have in case of plants. This is a disaccharide where alpha glucose is going to form a bond with beta fructose. Now you can see this is glucose, so this is the anomeric carbon, this is fructose, and this is anomeric carbon. So in case of this sucrose, this anomeric carbon is going to form a bond with this anomeric carbon. So this is alpha type, alpha 1 plus this is carbon number 2, because this is carbon number 1, this is 2, 3, 4, 5, 6. So alpha 1, 2, this is alpha. So alpha 1 to alpha glycosidic bond has been formed between the sucrose. 2 glucose, 1 glucose and 1 fructose is going to join together by V alpha 1 2 glycosidic bond. The important thing is both the anomeric carbon we have used. So if you have used both the anomeric carbon, we are going to call them non-reducing sugar. So sucrose is non-reducing sugar. The chemical name, because this yesterday I have told you this is pyranose ring. So this is alpha D glucopyranosyl. Alpha D glucopyranose ring is there. This is called alpha D glucopyranose, and when it is going to form a bond, we are going to call them alpha D glucopyranosyl. Which carbon to which carbon? Carbon number 1 is going to form with carbon number 2, and then beta D fructopyranose. Sorry. Now, one important thing in case of sucrose is called as invert sugar. Why it is called as invert sugar? If you will see the optical rotation of the plane polarized light, whenever, so in case of sucrose, the sucrose is having a plane polarized light of 65.5 degree right handed side. That is positive, means it is right handed side. When you are going to degrade it, when you are going to digest sucrose, you are going to get glucose and fructose. Glucose is having an optical rotation of 55.5 degree, and fructose is having an optical rotation of minus 92.5 degree. Means even if you are going to combine these two, joint these two, because both this is also 50%, this is also 50%, 1:1 ratio. So if you are going to combine, joint, add these two, so overall result will be in the minus. So when you have sucrose, a combined disaccharide, that time you had a dextrorotatory, means positive side of rotations, bun. When you have digested sucrose, you have got glucose and fructose. That time, because fructose is having more minus, so overall will be levorotatory. This is minus 92. So it will be, if you combine these two, it will be roughly minus 40, 42 around something will come. So overall optical rotation of these two monosaccharides will be minus 40 to 42 something will come if we will add these two. So sucrose have dextrorotatory. When you digest it, it will be levorotatory. So the optical rotation has been completely changed. Dextro becomes levo. So it is kind of opposite type of effect. So that is called as invert sugar. Because of this change in the optical rotation from the dextro to the levo, when it will be digested, we are going to call them invert sugar. Similarly, if you see lactose, in case of lactose also, it is also reducing sugar, because this is galactose, which is having anomeric carbon. This is anomeric carbon. So a bond is going to form between first carbon to the fourth carbon. So one of the anomeric carbon is left free. So bond is beta 1,4. Why it is beta? Because this is beta type. So beta 1,4 bonding will be there, and anomeric carbon is left free. So it is a reducing sugar because of this anomeric carbon has been left. Whenever you are going to digest it, you are going to get your galactose and glucose will be separated out. This is trihalose. this trihalose which we have seen previously, now you can see this is called as mycose or trimalose. In this case, this is glucose and this is glucose. This is first carbon and this is first carbon. So C1 of glucose is going to form a bond with C1 of glucose. So both 1 and 1 is going to form, means both anomeric carbon we have used for the bond formation. So it is non-reducing sugar. It is non-reducing sugar because no anomeric carbon has been left freely available here. Now this trihalose is chemically equivalent to glucose because if it will digest it, you will get glucose and you will get glucose. In our GIT tract, in the small intestine, we are having a specific enzyme that is called as trihalase. The specific enzyme which is present in my GIT tract in a small intestine, the name of that enzyme is trihalase. That trihalase digests this trihalose and gives you 2 molecules of glucose. So overall, if you are going to eat trihalose also, the ultimate product will be glucose. So it is equivalent to glucose. Effect will be, result will be equal to glucose only. And this is now this trihalose enzyme normally present in both in our case and in the other animals also, in the other herbivores also. So both places we are having this trihalose enzyme. So this is all about disaccharides. We have already seen how we are going to classify it, reducing and non-reducing sugar on the basis of which carbon we are going to use it. So if both anomeric carbon you have used it, we are going to call them non-reducing sugar. If one of the anomeric carbon will be left freely available, we are going to call them reducing sugar. Is it perfectly fine? Shall we go for polysaccharide or shall we leave it? It depends on you. Very small unit, a small thing is in the polysaccharides. Shall we go or shall we stop it? Mercy, after this one I will have your questions. Okay, anyone is having any question? If you have any question, you can ask me. In polysaccharide, we are not going to talk too much. We are having a small units only. It is a small things, so we don't have very much things. Okay, tomorrow. Mercy, anyone is having any questions? Mercy, you had a questions. So we will finish that question and then we will leave it. After this question, I will leave it. Okay. See this question. When we were talking about stereoisomers and diastereomers. Now, the question is which of the following is enantiomeric? This is your compound and you have to find out which one is enantiomer of this one. I will leave it. This question because Mercy has asked, so I will finish this question and then I will leave it. So first, whenever we are having any question on the basis of enantiomers and diastereomers, so first look at the carbon is minimum one asymmetric carbon you require there. If you are having any asymmetric carbon, then only it will have enantiomers. Diastereomers, forgot. Enantiomers is possible only when you are going to have minimum one asymmetric carbon. Now in this case, if you will say you don't have any asymmetric carbon because this is the central carbon, and what was the rule? Rule was first you have to look at the carbon should have 4 single bond. So this central carbon is having 4 single bond. single bond, single bond, single bond, single bond, and all those 4 things should be different groups. But in this case, 2 places we are having hydrogen. So when you have any 2 groups will be same. So this carbon is symmetric carbon, not asymmetric carbon. So in this case, because we don't have asymmetric carbon, so no need, means you don't have to go for finding out enantiomeric pair, enantiomers. So any of the things, it does not have any non-superimposable enantiomers possible. The question which you have asked is how to find out. So like this, you can see in this case, this compound and this compound, everything is same, only the location of the place of chlorine has been changed. In this compound means, if whenever you are having a solid line, it means the group is towards you. Towards you means upside. If you are having this type of a structure like dashed line, it means group is away from you. So this compound is different from this compound. Everything is same group, same place, but their 3-dimensional orientation is changed. 3-dimensional location has been changed. So this compound is different from this compound. Now we are going to say these 2 compounds. Mercy, now you have to tell me, probably it is not visual, you are not able to see it because of my picture there in the corner. So the compound is showing here, you have like this one, here you are having bromine, here you are having chlorine, here you are having fluorine, and you are having hydrogen. This is the compound. So if you will see the mirror, if I will put mirror, this compound, this carbon, the central carbon of this is asymmetric carbon. Why it is asymmetric? Because carbon is having 4 groups, 4 single bonds, and all the 4 bonds are different. Like hydrogen is different from fluorine, fluorine is different from chlorine, chlorine is different from bromine. Means all the 4 different type of group is associated with that carbon. Means this is having one asymmetric carbon. So obviously it will have a stereoisomers, it will have enantiomeric pairs. Now go and plot its mirror. So put it mirror and then draw its mirror image. So the mirror image of this one will be like this one. Clear? Now take this compound and put it on this one. If you will take this compound and put it on this one, will it be same or it will be different? This compound is both compounds are mirror image of each other, perfectly fine, but does it superimpose each other or not? It is not going to superimpose each other. Why it is not going to superimpose each other? Because this fluorine will come at this place, this chlorine will come at this place, bromine is at different place. So both the compounds are non-superimposable mirror image. So in this case they are called as, they are enantiomeric pair, they are enantiomer of each other, different. So it is not superimposable. So both are enantiomeric pair of each other. Now see this compound. In this case we have a carbon, but that carbon is not asymmetric, it is symmetric carbon. Why? Because 2 chlorine is there. So either chlorine is above or below. You cannot differentiate between these 2 chlorine. In this case we had 4 different groups are there. So this bromine is above the plane or below the plane, you can differentiate. But in this case you cannot differentiate which chlorine is above the plane, which chlorine is below the plane. Means both chlorine are of same molecules. So this case, this compound is, first this carbon is symmetric carbon. Why? 2 places chlorine is there. So if you will take this compound and put it on this compound, so this fluorine is on the plane and this one of the chlorine is above the plane, second component chlorine is below the plane. But you cannot differentiate between which chlorine is above the plane, which chlorine is below the plane. Means in this case you cannot call it this compound is superimposable completely to this compound. This compound is going to superimpose this compound. Why it is going to superimpose? This is diastereomers. Why? Because first you don't have any asymmetric carbon. You cannot differentiate between these two chlorine. So both chlorine are of same type. So here also if groups will be different, like how I can able to differentiate my right hand with the left hand? Because this is thumb. If all the 5 fingers will be the same, then I cannot be different. I am not able to differentiate my image with each other. Then it will be completely superimposed. Because this is my thumb and this is my index finger. So both structure will be different. If your all the fingers will be of same structure, same size, same shape, you cannot differentiate between right and left hand image. So right hand image will be left hand and left hand, then it will be completely superimposed to each other. So same thing going to happen in this case. You are not able to differentiate both the chlorine, so it will be superimposing on this compound. Is it clear? The main problem comes when you are having more than one carbon asymmetric. So that structure, that example we will come on another day. This is single asymmetric carbon compound I have find, so that minimum you will understand something, and on next day we will come with a compound which will have more than one asymmetric carbon, and we will have this type of structure, dashed and solid structures, and there we will discuss it. So for single compound, just find out it is asymmetric or it is non-asymmetric. If it is asymmetric, then it will be having enantiomers. If it is symmetric, it will not have enantiomers. It will be diastereomeric pair, or it will not have any optical isomers. Problem will come when you will have more than one asymmetric locations, places, carbons. So that structure we will talk slowly in the next day. Thank you. So thank you very much. Thank you. Any question if you are having, you can type, you can send, you can write. My mail ID, my phone number, everything is there. Whenever you want, you can contact any places. Okay. So thank you very much.